Hi everyone, in this video we are going to look at the heart muscle histology and the electrical conduction system of the heart. So this is video 2 of 3 videos for the heart chapter. So the last video we ended with a little bit of histology on what the heart muscle looks like, what is unique about the heart muscle. So you're looking at an illustration of the heart muscle fibers right here. So we're looking at heart muscle cells or heart muscle fibers. The plasma membrane here is called the sarcolemma. And the action potential here is going to start at that first pacemaker, which is on the superior end of the right atrium, which is your sinoatrial node. So a quick recap, the sarcomeres of the cardiac muscle cell look very similar to that of skeletal muscle cell in the sense that you have your M line. This is your thick filament. In yellow, the thin filaments are right here, exiting from your Z discs. And during the process of contraction, the thin filaments are going to slide over the thick filaments. So while the length of the filaments itself does not change, due to the sliding of the thin filaments over the thick filaments, the sarcomeres are going to shorten. So point to note is that the filament length does not change either for the thick or thin filaments. However, the sarcomere length changes. The sarcomeres shorten when there is a contraction of the cardiac muscle. Now here we are looking at an illustration of the SA node. The SA node stands for the sinoatrial node, and it is located on the superior end of that right atrium. So what you're looking at here, this amoeboid cell, is referred to as a nodal cell. Now, throwback to your nervous system and muscular system units from A&P 1, you already know the concept of RMP, or resting membrane potential. If you need a refresher on this concept, there is a video called Nerve Conduction where I go over what the concept of resting membrane potential is. Now here in your sinoatrial nodal cell, which is currently at rest, the resting membrane potential is negative 60 millivolts. So that means that the inside of the cell is negative 60 millivolts. So let's take a zoomed in look at this diagram on the right. So this is the plasma membrane. You're seeing the bilipid layer of your sinoatrial nodal cell. This is the interior of the cell where the cytosol is, which is the water component of your cytoplasm. And this is the interstitial fluid, also known as the extracellular fluid, which means that that fluid is outside your sinoatrial nodal cell. Now, you have some transmembrane proteins here. So if you recall with muscle contractions, there are 3 ions that play a role in muscle contraction. The first one is your sodium ion, which is Na+. The second one is the calcium ion, which is Ca2+. And third is the potassium ion, which is K+. So point to note that the sodium ions and the calcium ions, there are more of these ions on the outside of the cell. So on the outside of the sinoatrial nodal cell is where more of your sodium and calcium ions are going to live. Sodium and Ca2+ are highly concentrated outside the nodal cell, and there's less of these inside the nodal cell. Inside the nodal cell, you have more or a higher percentage of potassium ions. And action potential transmission is going to involve an influx of sodium inside the sinoatrial cell and an outflux of potassium outside the sinoatrial nodal cell. So here's the point to note. So sodium and calcium are going to go from out to in. Potassium is going to go from in to out. So this is how the electrical current spreads to the heart. So I want to recap the concepts here of systole, which means a state of contraction for heart muscle fibers, and diastole, which means a state of relaxation for the heart muscle fibers. So while two pacemakers, both the SA node and the AV node, can generate action potential simultaneously, we are going to break down and look at it in a chronological order, starting with the SA node up here on the superior end of your right atrium. So the SA node becomes depolarized, so it's resting at -60 millivolts. Sodium channels open, sodium and calcium influx into the SA node and depolarize the SA node. Now, that action potential spreads from muscle fiber to muscle fiber via the gap junctions. Remember, the gap junctions are located in your intercalated discs. So once the muscle fibers in this region are depolarized, those sodium and calcium ions can pass or permeate freely through your gap junctions and pass that action potential to the adjacent muscle fibers or muscle cells. So when that is happening, the electrical current, which is your action potential, is spreading from your SA node to the second node here, which is the AV node. Now the point to note is that action potential spreads slower from the SA node to the AV node. However, it passes faster from the AV node down to the bundle of His and to the bundle branches. So secondly, the action potential is delayed and it passes to the AV node. From the AV node, it is going to go through this AV bundle, also known as the bundle of His, which is located in that interventricular septum. From here, it is going to spread through the right and left bundle branch before it wraps around the apex and goes through the branches, which are your Purkinje fibers. So here, the AV bundle conducts that action potential and then spreads it to the Purkinje fibers, and all of your action potential, which is the sodium and calcium ions, pass through the gap junctions of the intercalated discs, and that is how action potential spreads all over the cardiac muscle in the atria as well as the ventricles. So here is the chronology of how that's going to happen. First, sinoatrial node depolarizes. That depolarization is spread over the right atrium and eventually on the left atrium. When muscles become depolarized, they contract. So this is when atrial systole will take place. Now this action potential spreads to the AV bundle, or the AV node. AV node depolarizing quickly spreads depolarization down to your bundle of His or your AV bundle, followed by the bundle branches and the Purkinje fibers. So as a result of this event, and this is an electrical event. As a result of this event, muscle on your ventricles become depolarized as well. So that is how both your atria depolarize from the sinoatrial node, your ventricles depolarize from the AV node. Now this can be studied, these electrical and muscular events can be studied using something known as the ECG, or the electrocardiogram. We're going to practice this interactively in class, as well as an assignment, an interactive assignment on McGraw-Hill Connect. So I'm going to explain this stuff in the diagrams that come here in the next few slides, but I want to go over what these terms in bold mean. So anytime you are looking at the terms depolarization or repolarization, this is referring to an electrical event, meaning that these events precede either muscle contraction, which is systole. So depolarization precedes systole, and repolarization precedes diastole. Now, depolarization and repolarization, again, are electrical events. Systole and diastole are muscular events. The next components are known as segments. So in segments, you will notice flat lines or plateaus on the graph. So plateaus or segments are where you're not going to see a lot of electrical activity, but you're seeing the flat or the plateau-like activity because a muscular event, contraction or relaxation, is what is going on during those times. So let's start with looking at two units of what you will see repeated on an electrocardiogram. So I'm going to draw a line here. So these are representing two separate cardiac cycles. So each cardiac cycle will be represented as a segment, followed by first electrical peak, which is represented as the P wave. Following the P wave is another segment or a plateau. The plateau that follows the P wave is called the PQ segment. We're going to talk about what events are happening at each of these segments in a minute. Following the PQ segment, you're going to see another electrical event, and that electrical event is a sharp peak. So that is your QRS complex. So the P wave is an electrical event. The QRS complex is an electrical event. Following the QRS complex, you have another plateau. The plateau is again this flat line right here, which is when a muscular event is taking place. The last electrical event here is the T wave, followed by another plateau before the cardiac cycle starts all over again. So I'm going to take the eraser and erase these scribbles so we can go over what this is one more time. So we're going to start off with the plateau, which has come which is the last part of the previous cardiac cycle. Now this is where the sinoatrial node depolarizes. This depolarization, which is an electrical event, is represented on this graph as the P wave. So the presence of the P wave means that your sinoatrial node is depolarized and your atria are depolarized, and they're getting ready to contract or systole. This followed by the PQ segment, which is right here, and I'm representing that in green. So this segment, the PQ segment, does not represent an electrical event, but it represents a muscular event. So what is actually happening here is the atria are contracting. So atrial systole is what is taking place within the PQ segment. Now, during this time, that action potential is spreading from the SA node to the AV node. So right here at the beginning of the QRS complex, this represents your atrioventricular node depolarizing, followed by that depolarization spreading down the atrioventricular bundle or the bundle of His, and your ventricles as a result getting depolarized. So your QRS complex, represents ventricular depolarization. So ventricular depolarization is when the ventricles are getting ready to contract. Following that is another plateau, which is your SD segment. So the SD segments is when there is ventricular systole happening. So your ventricles are actively in the process of contraction, and that is a muscular event. Now, after the ventricular systole happens, ventricles are getting ready to be repolarized, because after ventricular repolarization is when ventricular diastole or ventricular relaxation can take place. So the next electrical event is represented here with your T wave. Now this wave is representing ventricular repolarization, which is an electrical event. Now the plateau following your T wave, this little segment right here, that is representing your ventricular diastole. So let's recap this one more time, starting at the very beginning. This is when your SA node depolarizes, action potential is spreading over your atrial muscle fibers. That is represented by the P wave. P wave is an electrical event, meaning your atria are getting depolarized. Once the atria are depolarized, the PQ segment follows. The PQ segment is a muscular event, which is when your atria are in the process of systole or contraction. Next, the depolarization is spread to your AV node, and your AV node depolarizes, causing the QRS electrical complex. The QRS electrical complex represents the ventricular walls getting depolarized. Again, it's an electrical event. Following this is your ST segment, where your ventricles are in the process of contraction, which is your ventricular systole. Once the ventricular systole happens, the electrical event that follows is ventricular repolarization, represented in the T wave, followed by ventricular relaxation, which is the plateau that follows. Now the question here is, if you're seeing ventricular repolarization and ventricular diastole on this graph, why aren't you seeing atrial repolarization and atrial diastole on this graph, right? Now the reason for that is that event, atrial repolarization and atrial diastole, are happening concurrently as that QRS complex is happening. The QRS complex is shadowing on this two-dimensional graph. The QRS complex is actually shadowing the events that are happening concurrently. So simultaneously, as the QRS complex is happening, your atria are undergoing repolarization and atrial diastole is happening during the same time. So we will talk about the next few graphs here in class after we do the interactive assignment. So the last few slides here explain what I just explained on that one graph video. It spells out all the events, whether they're electrical, whether they're muscular, and what is and is not visible on your electrocardiogram graph. So in chronological order, first you have atrial depolarization, which appears as your P wave. And the start of that atrial depolarization at this point right here is where your sinoatrial node, your SA node, is depolarized. Next is your PQ segment, or the plateau, the atrial plateau. That is representing your atrial systole. Atrial repolarization and atrial diastole are not visible on EKG. Again, like we said, the QRS complex is masking that. The QRS complex is representing the electrical event of ventricular depolarization, followed by the next plateau, which is the ST segment, or the ST segment, which is where your ventricular systole is happening. The last event here, the electrical event, is your T wave, which is when the ventricular repolarization is happening, followed by ventricular diastole. So this slide talks about what those plateaus or intervals are and what their significance is. So the QT interval is basically time from the beginning of the QRS complex to the end of that T wave. So the cardiac cycle, so the next video I do will explain the entirety of the cardiac cycle. But the cardiac cycle involves both systole, which is contraction, and diastole, which is relaxation. So to transition into the next video, I think we all have to be familiar with that ECG cycle, what systole means, what diastole means, what electrical events are happening. Now, like we said in the lungs unit, right, in the respiratory system chapter, Boyle's law applies to heart chambers as well. So remember, Boyle's law describes the relationship between the volume of the container and the pressure inside it being inversely proportional. So contraction decreases the volume of the heart chamber, and therefore, by Boyle's law, pressure inside the heart chamber increases during systole. The opposite happens during relaxation or diastole. During relaxation, the volume of the heart chamber increases. Therefore, pressure inside it decreases. Just like air going from a high pressure to a low pressure environment, blood also moves down its pressure gradient. So blood moves from an area of high pressure, where there's more blood pressure, to an area where there's lesser blood pressure. That is where your valves come into the picture. So far we've reviewed 4 valves. 2 of the valves are atrioventricular valves, which is the tricuspid valve on the right side and the bicuspid or the mitral valve on the left side. We also looked at 2 semilunar valves. The pulmonary semilunar valve which opens from the right ventricle, and the aortic semilunar valve opening from the left ventricle. So I'm going to stop this video here, and we are going to pick up right here in the next video.